We've mentioned neurotransmitters in the past, but we need to go through a little more about what neurotransmitters are. And the cholinergic system, which basically is acetylcholine, they've actually named the entire system for one single neurotransmitter, but we'll go with it. So the cholinergic system is the first system to talk about. Now, this is acetylcholine. Acetylcholine is the first one that was really found. It's the most well-known one. We've already talked about it back in the neuromuscular junction as well. So here we're going to have both nicotinic and muscarinic receptors. So there's two different types of receptors for acetylcholine in your bodies. Now if you're thinking about it and saying, okay, nicotinic, muscarinic, it might sound a little familiar. Nicotinic, nicotine. That's why it's named. The nicotinic receptors not only bind acetylcholine, but the nicotinic receptors also bind nicotine. Muscarinic, well, it binds acetylcholine, also muscarine, which is a mushroom poison. So you have two different things binding to these receptors, hence the name of each one. Now we know it's released neuromuscular junction. Those are going to be nicotinic receptors. So that neuromuscular junction, nicotinic receptors. But we'll also see it at some of the autonomic nerve endings. So those motor endings. So you'll have these acetylcholine receptors throughout your body. So if you look at it again, released at the presynaptic terminal, this is that motor neuron coming down towards the synaptic. Diffuses across the cleft, binds to acetylcholine receptors. So think about on the muscle, for example. You bind those acetylcholine receptors. And then it's degraded by acetylcholine esterase. So all of those broken down components then are basically recycled back into your body. That is acetylcholine. So acetylcholine itself is looking at one of two receptors. The one we've seen already is based at the neuromuscular junction, but they both function pretty much the same way throughout your body. All these components, when you break down acetylcholine esterase, breaks down acetylcholine, are recycled. Your body is going to try to use as much as it can over again, because you cannot ingest enough if everything you utilize gets discarded as waste. So your body is really efficient at recycling things. All right, next category are your amino acid-based neurotransmitters. For these ones, looking at things like glutamate, you're looking at GABA, and don't worry about the full scientific name there, glycine. But these ones, the interesting part is they go through reuptake, which means they're pulled back into the actual presynaptic neuron after they're used. So it allows them to kind of be repackaged, reset, and used again. So you have this reuptake for these amino acid category. Next category is biogenic amines. Now, these ones are built off amino acids, but because they're using combination and putting them together, they're not just amino acids. Looking at serotonin, dopamine, epinephrine. And just like the amino acid category, these go through reuptake. They're pulled back into that presynaptic neuron to be used again. Neuropeptides, also based off of amino acids. But now these neuropeptides are a string of amino acids put together. There's no set length they have to be. All depends on what the signaling molecule is needing. These ones are degraded by peptidases. So think about an enzyme that comes in, just chops them up into smaller pieces. That way your body can recycle the amino acid components. So these neuropeptides. Now, all these different neurotransmitters can work in different ways, either a direct action or an indirect action. Direct action, you can see the actual signaling molecules are physically bound to the receptor. Channel opens and something goes through, whether it be ions or proteins or whatever. The channel opens directly. That's a faster, more direct mechanism, hence direct, because they bind right to it. The other mechanism is the indirect mechanism. Now, this is going to be coming up more and more in the next section in AP2 for the hormones, the endocrine system, but we're just going to have a little preview of it here. In the indirect action, you still have the signaling molecule binding, but it's not binding to the actual channel. it binds to a transmembrane protein. That signal triggers a G protein inside of the membrane. Now that G protein can cause different cascade of events depending on what has been triggered. But eventually, you'll get to these secondary messenger molecules. So it's not the same as the initial thing that bound. These secondary messenger molecules can then trigger the actual channel to open up. So you can kind of see how there's a couple of steps in between for this indirect action. So you have something binding still outside the cell that triggers a reaction inside, which has a cascade effect of triggering different things until you get to a secondary messenger. The secondary messenger then triggers the channel to open up. Still works, but there's a whole lot of other things going on in between. So they're direct or indirect mechanisms of how we're looking at these neurotransmitters working. So a couple different neurotransmitters, some different categories, and some mechanisms of how they're working.